Hey y'all, I'm Delana Moore, and this is a video on DNA replication, transcription, and translation for Dr. Mukhtar's genetics class. DNA replication occurs via the semi-conservative model. Prokaryotic chromosomes are circular and they have one origin of replication called the OREC and one termination region. Replication begins at the OREC and is bidirectional, forming a replication bubble and two replication forks. DNA topoisomerase plays an important role in this replication. It relaxes supercoils by breaking and unwinding the DNA. The broken strands are then rejoined by ligase. Eukaryotes have multiple origins of replication. They are also linear in nature, so the ends are synthesized using the enzyme telomerase. Thus, they are called telomeres. Let's take a closer look at replication. DNA replication begins when an initiator protein binds to the origin of replication. Next, helicase binds to the lagging strand and unwinds the helix by breaking hydrogen bonds. DNA gyrase relieves stress on the DNA molecule by allowing free rotation, and single-strand binding proteins keep the helix open. Initiation of synthesis requires an RNA primer made by primase. The primer provides a free 3' OH end for DNA polymerase III to use to begin synthesis. The leading strand is formed through continuous synthesis, however the lagging strand is made discontinuously by the formation of Okazaki fragments. Okazaki fragments are created when primase adds many RNA primers along the lagging strand. DNA polymerase then creates small fragments between the primers. These fragments are then joined by DNA ligase. All of the RNA primers are replaced by DNA polymerase I. DNA polymerase uses the complementary base pairing shown to elongate the molecule. All genes code for the production of RNA, which is read and formed through a process called transcription. The steps of transcription are initiation, elongation, and termination. The three regions used for this process are a promoter region, an RNA encoding region, and a terminator region. In initiation, RNA polymerase binds to the promoter region of the gene, and then the sigma factor binds to the RNA polymerase, forming a hollow enzyme. The DNA is then unwound to form an open promoter complex. The sigma factor is released, causing RNA polymerase to move along the DNA strand. RNA polymerase also uses complementary base pairing, but instead of thymine, it uses uracil. In prokaryotes, termination is either row-dependent or row-independent. Here, termination is row-dependent. A row protein moves along the new mRNA strand until it reaches the termination region. Then the mRNA is released. In eukaryotes, termination occurs in various ways. RNA processing also occurs. Modifications help the mRNA leave the nucleus and help with protection. RNA splicing is done to remove excess information from the RNA. SNRPs are markers to indicate the ends of exons and introns. Spliceosomes actually perform the removal of introns. Once RNA splicing is complete, the mRNA is ready for translation. Translation is the process of forming a protein from the RNA molecule that was formed during transcription. The three steps of translation are initiation, elongation, and termination. In initiation, the small ribosomal subunit binds to the Shine-Dalgarno box on the mRNA. Next, the RNA containing FMET binds to the corresponding codon. Then the large subunit binds, positioning the bound tRNA in the P site. A tRNA with the anticodon that corresponds to the codon in the A site will then bind. A peptide bond is formed between the two amino acids bound to the tRNA molecules. Then the ribosome moves down the mRNA. The bond between the amino acid and the tRNA in the P site is broken. As that tRNA moves to the E site, the tRNA then leaves. Now the A site is open, and another tRNA molecule that corresponds to the codon can bind, and the process continues till it reaches the termination region. Once the termination region is reached, a release factor binds and the complex dissociates, releasing the polypeptide. The difference in translation in eukaryotes is MET is used instead of FMET and Kozak sequence is used instead of the Scheindel-Garneau box.